Hi, MP Mark Warawa here, and I'm thrilled to be back at Trinity Western, where I went to university many years ago. And I'm with Dr. Chad Friesen, and you've just been awarded a discovery grant, and so congratulations.
Thank you.
It's very important. When you get a discovery grant, it really acknowledges the reputation that the university has internationally. Some of the brightest minds in the world are here doing important research. So could you tell us about what you got the grant for, what your research is?
Certainly. When I look at the research, I kind of divide it up into a couple of parts. So there's a hub aspect to the wheel of the research. So when we're looking at the hub, we're focusing on how do we make fluorinated salts from inexpensive refrigerant materials. Once we make these salts, then there are three spokes that come off the hub to establish the wheel of the research. One hub is looking at fluorinated salts to be able to make new polymers. One of the challenges when we're dealing with fluorinated epoxides, these are monomers to build interesting fluorinated materials. They typically are made in such a way that they only have functional groups on one end. So having a fluorinated salt would allow us to put tethers on both ends of the polymer so that you can do a lot more chemistry with the materials. So it would move it far beyond just tribology, which is protective surfaces for machinery parts. The other spoke that we are looking at is new personalized medicine materials for MRI. So what we do is we build fluorinated polymers that are vesicles that are quite small. So we're looking at an ability to put in drugs into my cells, and you'll be able to track that through MRI. The nice thing about that is when you're looking at these drugs, you're not adding too much to the patient. That's why it's personalized. So you can track where the drug is going to make sure it's going to its designated spot. And then you can also monitor how much is released over a period of time using MRI, but you need the fluorine to label those materials. So that's the second spoke. The last spoke of the three that are there, again, the fluorinated salt being the hub. The last spoke is looking at ways of being able to recycle precious metals. So when we're looking at catalysis, the challenge with catalysis is that it'll certainly do its job, but the question is how are you able to recycle those materials afterwards, and can you reuse those? So utilizing fluorinated materials, they can be used for recycling. So the nice thing about that is the MRI diagnostics that we are developing and the recycling of catalysts are very close, one and the same in terms of the concept. So that makes it quite interesting in that regard. And then hopefully the fluorinated salts, again, which is the hub, also will open us to a new avenue to do late stage fluorination of pharmaceutical drugs, because pharmaceutical drugs, about 20% of those that are currently made have fluorine in it. So we got to look at inexpensive ways to be able to put fluorine on them.
So very important applications to your research.
Yes, I certainly would agree to that.
Yes. Well, congratulations.
Thank you very much.
And I'm very proud of the work you're doing, and it's, as I said earlier, it's recognized internationally. So congratulations to Trinity Western University and to Dr. Friesen.
Thank you.